Okay, today's podcast is going to be on the major organelles that we study, and this is going to be organelle part 1. We're going to do the nucleus, the ribosome, smooth ER, rough ER, and the related one, the Golgi apparatus. Okay, first we're going to start with the nucleus. And the nucleus is known as the control center of the cell. And it's called the control center because that is where the DNA is going to be. Now the nucleus has 3 parts. The first part is the nuclear envelope. The nuclear envelope is a double membrane that surrounds the nucleus. And it has a similar function to what the cell membrane does. The cell membrane will regulate what comes in and what comes out of the cell. And so what this one will do is this will regulate what comes in and out of the nucleus. So once again, regulate what comes in and out of the nucleus. Okay, now an important part of the nuclear envelope is the nuclear pore. The nuclear pore is basically a series of proteins, and what that does is it is a protein, say a protein-lined pore that allows stuff to pass through and pass in and out. So we'll say just allows stuff in and out. Okay. So think of nuclear pore acts like a door. It allows stuff to come in and out of the nuclear envelope. The next structure that we're going to find inside a nucleus would be the nucleolus. This is a dark region, pretty much like a dark sphere. And what it does is it makes the ribosomes. And we'll talk about the ribosomes here in just a moment. The most important thing that's found inside the nucleus is going to be the chromatin. And if you can remember back from chapter 12, chromatin is DNA and its associated proteins. And those proteins, remember, are the histones. So remember things like a nucleosome, the beads on a string, etc. And if you could remember, DNA is the instructions for making proteins. And you got to have your proteins, especially the enzyme version of the proteins. All right, so let's draw ourselves a little picture. And what we're going to have here first is we're going to draw an outline of the nuclear envelope. And I'm leaving spaces for the nuclear pores. Now the nuclear envelope is a double membrane. So all these are going to be double. Okay, now we're going to draw the nuclear pores, and we are going to make the nuclear pores green. And so we're going to draw these proteins, and they form a tube. And remember, these are going to be the doors that allow you to get in and out of the nucleus. So like when we do transcription, the mRNA is going to go through the nuclear pores. Okay, the next item we're going to have in here in the middle, this is going to be our nucleolus. And this is the guy that makes the protein. And then we're just going to draw some yarny looking pieces in here. And this will be the chromosome and the DNA. Alright, so the red item right here, it would be DNA or chromatin. The blue item is a nucleolus. And then the black thing here is going to be the nuclear envelope itself. And we're going to call that NE for short. Okay. Now back over here to the ribosome. Back to our blue. Okay, the ribosome is a very small structure, and this is just a review of chapter 12. And really what the ribosome does is it makes proteins. So it's a little tiny protein factory. And remember, it's the site of translation. Translation is the process in which the ribosome is going to read mRNA and produce the protein. Alright, so let's draw a little picture of this. Remember, it kind of looks like a hamburger bun. And the way I draw it, there's 2 bites taken out of it. Right? So you got a large subunit, and then you got a small subunit. Remember, you got your P site and your A site. So just mainly remember the stuff from chapter 12. Okay? All right, the next thing we want to talk about is our two ERs. The ERs are membrane channels. All right? So anything that says an ER is going to be a membrane channel. And you kind of want to think of like a hallway or a highway system. So the ER is also referred to as the cell's internal transport system. Just a network of hallways, highways, roads. I've heard all different kinds about it. Now the smooth ER, the way you want to remember this one is it's called the smooth because it has no ribosomes on it. So if you look at it visually, it has a smooth texture. And it does a couple of things. And the main thing that it does is it makes and transports lipids. Not going to be able to make proteins or anything like that because it has no ribosomes. It also will contain, though, it will also have enzymes that are used to break down toxins. And you're going to find a lot of these in your liver. The liver's job is to break down many of the substances coming in your body into less harmful things. And one of those being toxins like alcohol, and also most of your medicines that occur. Okay, we'll go through there and be broken down eventually. That's why you would need to take the medicine again after 3-4 hours or so. Okay, now what we got here, we want to draw a picture of these. And one of the ways that you can tell a difference between the ER and the Golgi is that you got these little, it forms like a hallway, but then there is a channel or another small highway to the next level. Okay? And then, and it just kind of varies. They're not all perfectly symmetrical like this. Okay? So this would be typically what an ER looks like. And what would happen is that materials are made on the insides, and then they can be transferred down these hallways to where they need to go. Okay, the next type of ER is called the rough ER. And rough ER has ribosomes. And remember, ribosomes are going to be used to make proteins. So the function of a rough ER is to make and transport proteins. And because it's an ER, excuse me, we are going to draw a picture of it, and it's going to look just like this other one up here. So you got your little channels that come between them. But the one thing that's going to make it different is that we are going to have, ribosomes studying the surface of it. So you got all these ribosomes that are stuck to the outside of the membrane that makes up the ER. And this gives it a little rough texture, and this is why it's called the rough ER. And what will happen is that a protein will be made by this ribosome, and that protein is going to be able to travel down here to where it needs. Okay, so it's kind of like, think of this as like a classroom, and then when you guys leave the classroom, you go down the hallway to your next classroom, and this is the whole purpose of it. All right, a closely related structure to both the ERs is the Golgi, and we usually refer to him as our Italian friend who works in shipping, because he's actually named after an Italian scientist who discovered it. And the function of the Golgi is 3. It will modify proteins that come from the ERs. It will, modify. It's going to package those things. And they're always packaged in the vesicles. So it's going to make vesicles. And then it's going to ship them out. So think of like our Italian friend who works in shipping. So you modify, package, and ship, MPS, and send it on its way. Now, the Golgi essentially looks like a series of pancakes, and the pancakes come in different sizes. They start with a small one, and then the next one gets a little longer, and a little bigger, and then you got maybe the biggest one, and then they start to get a little smaller. So it's going to form this little kind of symmetrical shape. and then you'll usually see off to the side of it some of the vesicles that it's made. And these vesicles would have contained the things that were modified, repackaged, and then shipped out. Okay? And this will be able to deliver stuff throughout the cell and wherever it needs to go. Now, another organelle that is made by the Golgi is the lysosome. So you want to remember that the Golgi makes lysosomes. And lysosomes are pretty much little vesicles that contain digestive enzymes. And so a lysosome is going to do two jobs. Job number one is break down the food. So when this cell brings in any kind of solids through endocytosis, it's going to break them down. And we'll just call those biomolecules. Okay. The second thing that's going to do is, let's say you have a mitochondria or a ribosome or whatever. The lysosome is going to break down worn out and old organelles. And so this mitochondria that's worn out will be broken down, and the pieces will be recycled and reassembled into something else. Okay? And that will conclude our podcast on the organelles, part 1.